listen real close, because I cannot raise my voice. If I keep it at this level, I might last 2 classes. That's what I'm done with that. Okay, so you hadn't asked questions all year, and today you asked questions.
We just started. So if your voice gives out, you know, do we have to come back?
Yes.
You have to remember, there's no stupid question except when you don't ask.
That's a matter of opinion. We left off right here at endocytosis. We're gonna see more of this in chapter 6. But engulfing inside the cell. So really what happens if something inside the cell is not supposed to be there, or it doesn't function anymore, then lysosomes break this further down. And as they break it down, we have to bag it up, trash. So large particles, phagocytosis, take care of it. Small particles, penocytosis. And we have this is literal definitions right here with cell eating and cell drinking. So we bag it up, we set it out next to the cell membrane, and then we push it past the cell membrane called exocytosis. Basically setting garbage out on the curb, what it is. And animal cells, they're called vesicles. The garbage cans are called vesicles. So that's all that is. We'll see that again in chapter 6. There's exocytosis right there. Quick to the point. Cilia, locomotion. In the human body, you'll find it in the respiratory tract. You'll also find it in the fallopian tubes of females. It is to move something past it. Flagella, one place you find it in humans. Everybody will take a guess. Oh my gosh. Where would you find flagella in humans? Sperm. It's only, reproduction is a lot closer than intestine. Microvilli increases surface area of the small intestine. There's also villi there as well. They serve 2 different purposes, but they're still a little bit similar. So.
Where'd you say cilia was?
Fallopian tubes and respiratory tract. The villi's purpose is to absorb fats. They're much larger, there's fewer of them, but they absorb fats. Doesn't make a lot of sense to me that they absorb fats. And then that goes to the lymph system. Lymph dumps it back in the blood system to clog your vessels. So that's where it goes.
I shut that door.
Sure. Yeah, we don't want Connor in here anyway if he shows up. All right. Cytoplasm and organelles, that's a liquid portion of the cell. All the little tiny organs, we're going to hit the highlights. Lysosomes are the police of the cells. Suicide sacs is another one. Garbage disposal cells. They're the ones that come in and clean up the mess when something doesn't work. Autophagy is a process that destroys worn out organelles so they can be replaced. Apoptosis is programmed cell death. In other words, your cells are programmed to live for so long, then they're going to die. And they've done tons of research on this. We can't change that genetics. You're going to age, you're going to get old, and you're going to die. That's what all the research says. It doesn't matter. Here's a test question. Top statement. Cells cannot undergo apoptosis without lysosomes. We need the lysosomes to clean up the mess. A close cousin, the lysosomes, peroxisomes, they use a little different enzyme. One uses a proteolytic enzyme, the other one uses hydrolytic. I think lysosomes uses hydrolytic and peroxisomes uses proteolytic. Not that it matters. One can help each other get that job done. Mitochondria is the powerhouse of the cell. Wednesday, when we do cell respiration, you'll understand that. Endoplasmic reticulum, there's rough and smooth. Rough has ribosomes. What's the purpose of a ribosome? We just put it on the board Friday.
Ribosomes, it's creation of new cells.
Protein synthesis, yeah. Create proteins right there on the ribosomes. That's the rough. The smooth deals with detoxification and lipid synthesis. You'll find a lot of smooth ER in the liver. That's what the liver does, is detox. That's one of many things it does. Golgi complex packages and ships proteins. It does other things, but this is one more known things that it does. And so if you look back, you've got lysosomes as police of the cell. You've got the mitochondria as OG&E of the cell. You've got the endoplasmic reticulum as the emergency room of the cell. And Golgi complex is the postal service, except this will get it there on time and it won't be broken. It's supposed to be funny, it's the best I can do today. You're not awake at 9 in the morning. You should have taken my 11 o'clock class. You're a lot more spunky at night when you took that night class. Cytoskeleton, made of two different kinds of protein. Tubulin's the larger of the two. Actin is the much thinner, thread-like, structuring the internal part of the cell membrane, so that cell has a lot of flexibility. Yeah, we finally made it where I wanted to be today.
You're slacking on us.
I'm ahead in the 11 o'clock class, which I'm supposed to be, but I can't remember where I shut them off and where I got us shut off. We should actually get to where I had them today for us, because they're labs today, so they won't get a lecture.
Our lab's today.
This afternoon, 11 o'clock is the 11 o'clock lab. Y'all have 2 o'clock, so. Is that too confusing?
You just got all kinds of fun today, don't you?
Yeah. That's why I didn't want to miss. I'm so dedicated. Why did they get me to stay home? Those kids will be so mad at me. She just swore to that too. All right, the zygote result of union of egg and sperm. So we're going to talk a little bit more of that as we go through this.
That's
Now,
question.
possibly. If we compare mitosis and meiosis, we see mitosis still in normal, everyday body cells, where these cells produce little clones of each other. If there's 46 chromosomes, we produce cells with 46 chromosomes. That's just what happens. So 2N is diploid, and that's 46 in humans. Meiosis deals only with sex cells. Haploid, we start as a 2N cell, cut it in half. As an N cell, we have 23 chromosomes. And that's important because if mom has 46 chromosomes and dad has 46 chromosomes, egg and sperm unite, we've got 92 chromosomes. Is that a human?
No.
Does anybody know what that really is? Kelsey? What? It's a goldfish, thank you very much. I'm so proud somebody from my Zoe class remembered. 92 chromosomes is a goldfish. Humans don't make goldfish. If you remember from an early zoology lecture, we have in there that poodles make more poodles. Remember that? Chickens make more chickens, therefore humans make more. That's why you made an A in that class, right? All right, so we have to go through meiosis, cut that in half, cut mom in half, and those two join, we'll have 46. So meiosis is the key to reproduction, sexual reproduction, okay? Now, as we get into the cell cycle, the cell cycle incorporates interphase, prophase, metaphase, anaphase, telophase, incorporates all of them. What we were doing Friday, showing replication, transcription, and translation, all those are happening in interphase, okay? I believe it's page 72 in your text. If somebody has a text there at your desk, flip that open, both of you can look at it. It's not 72, maybe it's 73 or 74. We're looking for a pie chart. There it is.
74.
74. My books start running together after a while. Notice in that pie chart that most of that is an interface. You got G1, S, and G2. So I'll probably ask you, what are the parts of interface? I may ask you, what part of interface is DNA replicating itself? That's the S phase. It always has been and probably always will. Now, it depends on what book you read, what's really going on in G1 and G2. I think all of them agree there's final growth, but some say there's some protein synthesis in G2 as well. But there's a ton of stuff going on here. We're doubling all the genetic information. So when we get it doubled, we can now start organizing and dividing the cell. And that's all mitosis is. So if I ask you on the test, what are the phases of the cell cycle? You're going to start with what phase?
Interface.
Interphase, and then go through mitosis. If I ask you just the phases of mitosis, you will start with prophase. So we need to know what goes on with each one of these.
Now question.
Okay?
If you want the stages of cell cycle, do you want us to break down interphase, or do you just want the 4 phases?
The question probably just asks what are the phases, and you start with interphases, the cell cycle. And I may have other questions about what's going on in these, or may say, what are the phases of interphase? What are the parts of interphase? Something like that. So
Now
it's
here's-
important that we understand that most of the cell cycle is interphase. I mean, it takes a while to get her act together. It doesn't take long to organize and separate, but getting her act together is what takes a while. So prophase, centrioles move to opposite poles, nuclear membrane disappears, and chromosomes appear. Metaphase, chromosomes line up. Anaphase, chromatids separate. What is a chromatid? I can get y'all to talk and just say my voice is old. Chromatids, a half a chromosome. Chromatids are made of DNA and proteins.
Half a chromosome.
Half a chromosome.
Made up of DNA and what?
Proteins. So technically, chromatids are made of chromatin. Chromatin is DNA and proteins. And 2 chromatids make a chromosome. That clear that right up for you.
Mhm.
So when it says the chromatids separate and move to opposite poles for anaphase, what it's saying is that these chromatids are sister chromatids, also known as homologous chromatids. Those 2 terms mean they're alike. In other words, sisters would be, how many of y'all have sisters in here? Do you look just like your sister? Do you act just like your sister? Are you pretty darn sure you came from the same genetic pool? Some of you a little skeptical there. So you're not sure you can. But we wonder that. But ultimately, is what we're saying is that these sister chromosome or chromatids are sisters. In other words, they carry all the genetic material. Would you say your sister has hair and eyes and ears and teeth? And you have hair and eyes and ears and teeth, maybe different color, right? But at least you have it. Okay. So that's the same thing here. This, if that's a chromosome and chromatids separate and move to opposite poles, we're going to the poles here. Each one of these are sisters. They may carry blue eyes, brown eyes, but they're still carrying all the information they need. Once they reach the poles, that's when telophase begins, and that's when they become chromosomes. When the chromatids reach the poles, they become chromosomes, and that's when telophase begins. Now, when do we see telophase? We see it when the furrowing effect starts. When you see this pinching apart, that's how we know when we visually see telophase. That's how we can tell. If you don't see the furrow, we're not in telophase yet visually for us. I'm going to show you some of those in just a minute. Cytokinesis happens same time as telophase, and that the division of cytoplasm is going to happen then. Necrosis is tissue death due to cell death. Hypertrophy is growth due to increase in cell size. That would be basically us lifting weights. If a small child is growing, he starts this tall and he ends up this tall, that's hyperplasia. That's increase in cell number. There'll be a point at which you quit growing, as we know. But what age does girls quit growing?
I was 12.
12?
That's not wrong.
Me too.
You know, really should grow till you're 16 or 17, maybe 18 at the far end. We got some genetic phenoms. When does guys quit growing?
Usually 18 to 20.
Well, I'm 47, I haven't quit growing yet, I'm just growing circumference now.
Height-wise, 18.
Height-wise, 18 be the low end, all the way up to 21. I was one of those that I grew about 3 inches between 18 and 21. So I've grown about 10 inches since then.
Circumference
All right.
varies.
Do I?
Circumference varies.
Yeah, certainly. This reference here, I'm not sure it's actually page 24 in your text, but it was in some text somewhere at one time. But the P53 and P21 genes, this is what they discovered control mitosis, these two genes. And they control the substance called cyclin. When they allowed cyclin to be produced, mitosis happened. When they stopped cyclins, mitosis stopped. And in the research, what they found out is if they went in and with x-rays and bombarded the P53 gene and killed it, and that laboratory animal that they killed the P53 gene in got cancer. If they went in and killed the P21 gene, that animal got cancer. If they killed both or either, the animal got cancer. So mitosis, or excuse me, cancer is mitosis out of control. These two genes control cyclins. When they were gone, cyclins ran rampant and mitosis ran rampant. And it was out of control. So we'd create so many cells here, they had a place to go. So they go somewhere else and attach and start making. Go somewhere else and attach and start making. That's all cancer is. It's cells where they're not supposed to be, randomly running out of control.
Page 74 starts for P53, but there's no reference to P21.
In the text. I have the liberty of making stuff up as we go as well.
They just forgot to add it.
I'm
from
sure I've
one
read that
transcript.
somewhere. I think it's in, actually, the P21's listed in the Zoe book, actually. So if we look at this, we have animations, but we also have actual pictures up at the very top of the interface. And a little difficult to see from the back. Let me see if this will help any at all. Can't really tell much, can you, on that first picture? See a little dark inside there, a little dark area? You can see the nuclear membrane around it. That's interphase. If we get here, nuclear membrane's disappearing. I don't really see a circle around it. So that's an early prophase, much darker, much more condensed. Later prophase, much more organized, getting there. We drop here, we're organized in metaphase. We're lined up in the center. Then in early anaphase, you can barely see breaking apart right here, early anaphase. And then we're really moving opposite poles here in that anaphase. The telophase, can you see at the top the cleavage, the furrow? At the bottom, you barely, I can't really make it out at the bottom, but you can certainly see it at the top. And so in telophase, we're dividing out the cell. What happens simultaneous to telophase? Division of the cytoplasm. What is it? It's in your notes. Starts with a C. Cytokinesis thing. And so when we're finished with telophase, we produce two daughter cells just like the original mother cell. And we start it back over. Now, does it start back over? It depends on what part of the body we're in. In the stomach, it replaces itself every 2 to 3 days. Your skin, 2 to 3 weeks. So if it runs real fast, depends on where it's at. Runs a little slower, depends on where it's at. But it's all controlled by those 2 genes and how much cytokines that it allows, produces.
When does crossing over occur?
I haven't got there yet, but I'm getting.
Okay.
Been working on study guide, huh? Very good, I'm impressed. This is lab. This is what we'll look at in lab. And what you have here is an early prophase. In early prophase, you see chromosomes. This is all chromosomes, or really it's chromatin. It's not organized. We're not joined up. We're not condensed. We're not coiled. Very early prophase. Is this one much more organized? I can tell it's more organized. And you can almost see chromosomes. There's a chromosome, there's a chromosome, there's a chromosome. And so this is a later prophase. Each stage we're getting more organized. We're going from that to a much more organized metaphase. You can see the 2 centrioles. Chromosomes are lined up in the center. From there we'll go to anaphase. You don't see a furrowing effect here, but we see chromatids separating, moving opposite poles. And from there, we go to telophase. You can obviously see that's telophase. So when we get to our lab today, we're not gonna go over mitosis, I just did. What stage is that? That one? That one? That one? Which one?
Late
Prophase? Early prophase. Now, don't do like some people do in my lab, and I'll put that down, they'll say mitosis. 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 That's, yeah, it's mitosis. Which stage? Metaphase. These pictures are out on my website, so you can study them in as well. Now, we want to finish up with our comparison to meiosis. Now, mitosis, we're producing clones. Meiosis, we're cutting the number in half. Here's your definition of meiosis. You double the number of cells, half the number of chromosomes. Double the cells, half the chromosomes. Meiosis. Double cells, half the chromosomes. So we look at this animation and try to simplify because they just give us 4 chromosomes to work with, and that's good for us. We get to using toes and fingers both. It gets smelly here. So we start with the first one. We're in prophase 1. You see 4 chromosomes, 2 red, 2 blue. Then as we continue through prophase 1, we see different things happening. We see synapsis. Synapsis is joining of like chromosomes. That's all it is. We're getting more organized each stage we go. So we join up like chromosomes. And then we start the crossing over process. And that's what you asked about, wasn't it? Crossing over? Crossing over occurs in prophase 1 of meiosis. That answers that question. What is it? It is the exchange of genetic material. You see these, we was talking about sister chromatids earlier. Well, we have a bunch of sister chromatids here. These are actually sister chromosomes, which means all the chromatids are sisters too. Also known as bivalents, tetrads, or homologous chromatids, like we said earlier. And tetrads mean there's 4 sisters in this deal. So, when they cross over, we see part of the blue one exchanging genetic material with part of the red one, and vice versa. And only in meiosis and sexual reproduction does this occur. Its advantage is added diversity to the offspring. So where this set over here may have gave you blue eyes, after they exchange material, we might have got green eyes or brown eyes to go with different colored hair. And we exchange over here, so you can see all the different combinations that we can throw together here.
Question or statement. Doesn't it also only in meiosis do a random mix? It doesn't do the same mix every single
Oh,
time?
it's random. Yeah, that's some of Mendel's genetic laws that he put together. And everything's random. And every child comes from the same gene pool. You throw them all in there and you dig out 46 chromosomes. Next child, you throw them all in there and you dig out 46 chromosomes. Combinations can be so unique. If you like one combination and you try to get it again, you probably won't. In my case, I didn't like any of the combinations. We continued trying and still hadn't found one we liked. So, it's pretty sad, isn't it? We just finally got too old to quit. So as we synapse, we cross over, we exchange genetic material. Here we see we're lined up. But notice we're lined up as paired chromosomes. In mitosis, we were lined up as single chromosomes. That was mitosis. In meiosis, we're paired up as homologous chromosomes, or tetrads, or bivalents, however you want to call that. And then we get here in anaphase, and the chromosomes separate, move to opposite poles, and we divide the cell in telophase. We take those two and start meiosis 2. Now, why do we need to do meiosis 2? How many chromosomes we have in each cell?
23.
Well, in this one 2, but yeah. 2, we started with 4, so we already have the number of chromosomes, but what have we not fulfilled? What's our definition? Is my voice freaking you out that much?
We haven't fulfilled the diploid.
The double the number of cells.
Yeah.
That's what we have, so that's what we're doing now. So we take these two, we're not real organized here, but we get organized in metaphase. Anaphase, chromatids, separate, move to opposite poles, because they're all sisters. Then telophase, we end up with 4 cells, not 2, like you get mitosis, but 4 cells. How many chromosomes do we have now? Still have 2 going. So we've half the number of chromosomes and double the number of cells. Now, this is important in meiosis. If this is sperm production, these would be 4 equal sperm. If this was egg production, there'd be 1 good egg there, and it would be big. And you have these little bitty polar bodies. Have 3 polar bodies, which are non-functional eggs. If you think about it, if all 4 of these were equivalent eggs, are we made to have 4 at a time? I mean,
It's possible.
it's not normal for humans to have 4 at a time. If you have 4 kids, we're gonna say, what drugs were you taking?
Mhm.
There'd be some fertility drug probably involved in that. Because it's not normal. So if we go back to meiosis 1, what you end up with in meiosis 1, instead of 2 equal cells, we have 1 big egg and 1 little egg. The genetics is here, but but the cytoplasm's not. Then we go through meiosis two, and this one divides out into two more small eggs, and this one divides out into another small egg, and so we end up with one big egg and three polar bodies. Why? Because we need the cytoplasm all in one cell, because that has more mitochondria in it for the energy necessary to sustain life after fertilization. If they all had equal amount of cytoplasm and they all got fertilized, they'd all starve to death. So we concentrate all our energy in one. We couldn't handle four anyway. We'd go postal on something. So that's why in egg production. Now, we do have twins. There's a lot of twins. How do you explain twins? There's two ways.
You started out with
You know what
two.
twins are?
You started out with two instead of one.
Two eggs, two ovulations, so you'd have two good eggs and you'd have 6 polar bodies. And that could be a boy, girl, or non-identical, what do they call them? Fraternal?
Fraternal.
Fraternal twins. And identical twins are same egg fertilized, split after metaphase 2. Metaphase 2 is supposed to be the point of fertilization. And then when we finish that, in fact, meiosis 2 does not finish, fertilization does not occur. So when fertilization occurs at meiosis 2, and then it doubles material and splits again, then we have identicals. And they'll always be of the same sex. So typically in identical twins, you can have 2 different placentas, or you can have them in the same placenta. If you had your choice, you want 2 different placentas. In fraternal twins, you almost always have 2 placentas. Not always, but most of the time you would, simply because we're dealing with 2 different eggs and they attach in 2 different places and develop their own placentas. Any questions there? Feel educated today?
Your voice needs a break. We'll stop here, right?
It's starting to break, isn't it? I'm gonna save it for the next class. Lab today, we're gonna work on 1.1 and 1.2. We're gonna go over tissues. We've already done mitosis. And, when you turn in 1.1 and 1.2, before you leave today, we'll be finished. We'll do chapter 5, cell respiration Wednesday and review. It won't take very long to do this. We'll review. If my voice is strong and we get done quick enough, we'll be in chapter 6 as well. So,
Don't confuse us before we tell us.
I'll see y'all this afternoon.
Where's lab at?
Come get away, Joe. See you this afternoon, 2 o'clock.
Michael.